Hey everyone, my name is Gabby and I will be discussing optogenetics and its applications. So, for this video, we're going to be discussing the definition of optogenetics and how it occurs, as well as how it's used in neuroscience, more specifically in current research. So, what exactly is optogenetics? So, it is a technique that is used to alter neural activity with the use of light. It enables optical modulation of selected cells with the use of engineered proteins. So in this case, the light activates these engineered proteins. It's important to note that opsins are light sensitive proteins that function as ion pumps or sensory receptors, and that they can be found in basically all organisms. Microbial opsins capture light energy and they use it to actively open channels, which then allows for ions to flow across cell membranes. So, how exactly does this work? So, we see the on and off switch, blue light and yellow light. And so, based on what I know other research studies do use different colored lights, but for this example, we're going to use blue and yellow. So, under the on switch, we see that blue light activates the on opsin, which allows for positively charged ions to enter the neuron through this opsin, which then results in the neuron firing. And when the neuron fires, the neurotransmitters are released, and this allows for the function to occur. And on the other hand, under the off switch, when the yellow light activates the off opsin, negatively charged ions enter the neuron through that opsin, which then stops the neuron from firing. And when the neuron does not fire, the function therefore does not occur. Now I'm going to show you various applications in current research involving optogenetics. The first application is a study conducted by Luo et al. In 2019, which used photoelectric fluorine-bound photosensitive proteins, or basically just HEBR, in order to reprogram human fibroblasts. So researchers transfected, or in other words, artificially infected or introduced certain genes into the cells, into human fibroblasts using fullerene. And so, what exactly does this mean? So, previous research has shown that environmental stresses such as acidity can stimulate cell reprogramming. And so, when they did this, they found that the pH in and out of the transfected fibroblasts changed a lot. And so, this change caused the fibroblasts to differentiate into neuron like cells. So, basically, the fibroblasts turn into neurons, and this can be applied in a clinical setting, especially for neurodegenerative diseases such as Alzheimer's, Parkinson's, Huntington's, which are commonly associated with nerve damage. The second application I have over here is a study conducted in 2018, which used optogenetics to achieve far red light to control the expression of genes. So, what researchers did is that they induced pluripotent stem cells into functional neuronal cells. This allowed for gene editing, more specifically in the enzyme called Cas9, which is deactivated. This enzyme basically acts as a molecular scissor function that allows for transcription to occur. So this really signifies the effects optogenetics can have on gene editing in particular, especially when it comes to DNA. The next application I have over here is a study conducted by Lee et al. In 2019, which used optogenetic techniques to inhibit pre-LCPDYN neurons, which are commonly associated with neural circuits that regulate sodium appetite. So, what this means is that this can affect one's appetite for salty foods. So, this can be particularly useful given that we know that salty food um, can lead to high blood pressure and heart disease as well as stroke. So this can really help modulate that. The last application I have over here is a study conducted by Weber et al. In 2015, which used optogenetics to demonstrate that the activation of GABAergic neurons in the ventral medulla can reliably induce REM sleep and prolong the duration of REM episodes in the mice. So what the study basically proposed is that they were basically turning REM sleep on and off, and they were able to keep mice in that REM stage for longer. So, the ability to control REM sleep at a high temporal precision, as demonstrated in this study, should provide a useful tool for the study of its functions. And we all know that REM sleep can help us regulate our emotions and reframe negative experiences through the process of dreaming. And so, future applications of this research could be potentially useful when it comes to aiding people with sleep disorders and issues that require them to demonstrate higher REM sleep. So that is all I have for this presentation. I hope you enjoyed and learned a lot. Here are my references, and if you have any questions, feel free to reach out.